All right, well, let's get started. Today, we want to spark some new areas of the brain and have your brain secrete that stuff that it does when it's making new neurons. And we want you to look at these things a little bit different, a little bit deeper. We're talking about the first study I'm talking about has to do with the X39. We call it stem cell activation patch. But the study and the effect of the patch has everything to do with the actual peptide known as the GHK copper peptide. So I'm calling it the GHK copper peptide study. The research here, you can go back and read these. I'm not going to read them right now for time's sake. It was published March 7th, 2021 in the Internal Medicine Research volume. I got to get this out of the way. You'll have to go back and read that. I couldn't see it because of your pictures. So this study was a double-blind, randomized controlled trial that explored the impact of wearing the X39 transdermal photobiomodulation patch over a period of 1 week on the level of GHK copper peptide. Now you see the study. We want to show if wearing this patch elevates that peptide. Here's the cool thing. This research paper was presented to the U.S. Patent Office in order to receive the patent for the X39 technology. That's how groundbreaking and important it was. So this is the process. Started up here, 60 people were randomized into two groups. 1 tube of blood was drawn. More blood was taken at 24 hours and 7 days. The result, this is like an overview, a significant increase in GHK was found compared to the control group. So the X39 was worn for a total of 7 days, and they looked at the increase. That's what they were proposing and asking, will it increase the GHK copper peptide? So in order to understand what the patch does, let's turn to PubMed, National Institute of Health, and look at some of the studies that have been done on that peptide. Hold on to your hats. First isolated, 1973 by Dr. Lauren Picard. David Schmidt talks about him very highly and says he should have received the Nobel Prize for the discovery. Maybe he still will. It belongs to a group of what are called emergency response molecules that your body puts out during injury to come to the body's aid. Hey, I got something going on. I'm going to have to repair. I'm going to have to rapidly heal some wounds. I need this, I need that. That's what that GHK copper peptide does. It's naturally sent out to those injured tissues. It has been shown to have a therapeutic role in age-related neurodegeneration and cognitive decline. This is one of the reasons we use them in my practice, which is we help people with brain issues. Improves axon survival. I'll explain that in a second. And maintenance of nerves. We've heard testimonials of people who get great benefit when they have neuropathy, diabetic neuropathy, and other issues going on with nerves. And the axon is the big long part of the nerve, and it helps maintain those and maintains the nerves. Let's see, is my screen frozen? No, okay. So let's talk about some of the actions of the GHK copper peptide. Activate stem cells, that's what we talk about. But that's only one category that this GHK, of the effect that the GHK can have on the body. Accelerates the speed of wound healing. Beneficial for hair, skin, nails, both growth and repair. And here's some subcategories. Tightened loose skin and thickened older skin. Repaired protective skin barriers. Who doesn't want to have the younger looking skin as we age? improved skin firmness, elasticity, clarity, reduced fine lines, depth of wrinkles, and overall appearance, and smooth, rough skin. More actions of the GHK. I don't know if you've heard this yet, but it's an antioxidant and it's anti-inflammatory. It beneficially altered the expression of human genes, and I'm stating it in the past tense because these are according to studies, and this is extracted from PubMed, National Institute of Health site. Beneficially altered, so genes will either tell the body to do something more or do something less. And so it was about 50-50 activating the genes and to do what they are designed to do. Some of them go up and some of them go down. Induced strong anti-anxiety, anti-aggression, and anti-pain activity within 12 minutes in a study of rats. This is why people in my practice say, wow, I just feel calmer, or this pain went away. Alleviated neuronal apoptosis and intracerebral hemorrhage. Let me explain that. If you have a head injury, you get hit in the head, and you'll have swelling or hemorrhage, bleeding inside of the brain. If you have ample GHK copper peptide in your brain, in your blood, the number of neurons that will die decreases dramatically, which is huge for maintaining yourself, your brain function if you're injured. More actions. It's been implicated in the resetting of 4,192 genes to a younger, healthier state. We talk about that all the time. It resets, you'd be safe to say, about 4,200 genes to a younger, healthier state. It is the #1 bioactive compound recommended for the treatment of COPD. I want to make sure everybody knows this is according to research. I am not making any medical claims, but it resets the programmed cell death cycle in different types of cancer cells. So why are cancer cells cancer cells? Well, they don't get the message when they're sitting there in some tissue. Hey, I got a neighbor on the right, a neighbor on the left, a neighbor in front, a neighbor in back, a neighbor above, a neighbor below. Hey, I don't need to divide anymore. Cancer cells don't get that message. They just keep dividing, and they invade the neighboring tissues, and that's why cancer can be fatal. Well, the GHK reset that cell death cycle in several different types of cancers. It has never been shown, the GHK copper peptide in all of the research since 1973 has never been shown to cause any side effects or issues. So if someone comes to you and they've been using the patch for a day or two or whatever, and they say, hey, this happened. Well, the first thing you want to say that there are no side effects from the GHK. It's your body's inability to do something like detox or to perform something that you haven't been really healthy and you have compromised detox pathways. That's usually the cause of that. It's not a side effect of the GHK copper peptide increase in your blood. More actions. It's being used now to heal spinal segments. And what they do is they do subcutaneous injections of the GHK copper peptide in over areas of the spine that have been damaged, and they've actually been able to heal discs and things. Well, we don't do injections, do we? We just increase the GHK copper peptide systemically, so they have ample in the brain, in the spine, in the kidneys, everywhere in the body, rather than a small injection of a 1/4 milligram of GHK copper peptide and subcutaneously. This is a little confusing. I'm going to explain this. Enhanced activity of alginate hydrogels for human cord blood, mesenchymal stem cell osteogenesis. Osteogenesis is the creation of bone, new bone where it needs to be created. alginate hydrogels, they put this stuff on areas so that it will stay there when they want to cause something to they want to heal something. Well, the GHK enhanced this bone regeneration in these alginate hydrogels, increase the healing speed of many tissues. And we hear these testimonials, you wonder, how in the heck is that happening? Well, it increases the healing of skin, stomach, intestine lining, and bone tissue, to name a few. Few more. You're going to see some slides in a minute that is very pertinent to this. Strongly chemoattracted, I'm going to explain it, capillary cells, mast cells, and macrophages. Chemoattractive means, hey, where it says capillary cells. So they are going to infiltrate tissues where they need to go. And it's the GHK that says, hey, over here, come and get me. And then the capillary cells chase it into the damaged tissue. And you get rapid growth of these capillaries. When you have big cuts, lacerations, injuries, there's no blood flow. There's no blood supply. So the GHK strongly chemoattracted those things in immune cells, macrophages. They go in and gobble up the bad guys. And mast cells, that's part of the immune response as well. Strongly activated genes of the ubiquitin, here's some more fun words, proteasome system. So we have a system of communication in our body, so lots of them. One of them is to send out some kind of a chemical, a signaling molecule, kind of like the GHK, but it only needs to turn something on and stop. It can't keep doing the thing that the body's telling it, that your body's trying to tell those whatever it's doing, and so it has to be cleaned up. The ubiquitin proteasome system is used for cell cleansing. So you think about that, cleaning up stuff that we don't need anymore, how that can also promote good health. Here's a biggie. When someone says, hey, I had stem cell injections and they didn't work. And when you know that stem cells are attracted to inflammation like a moth to a light, but if the inflammation is too great, it's more like a moth to a flame and they die. This is why the doctor has to say, hey, they might not work. It's because of the inflammation that they wouldn't work. Here's why they can't give you cortisone. Cortisone has anti-regeneration properties. So they're not going to give you cortisone to cut down the inflammation. This is why it's so great with the Aeon and the X39, because the Aeon reduces the inflammation naturally without cortisone, but the GHK helps block the regenerative effects of cortisone. Last two biggies, especially for me, not that I'm special, but I've had huge changes in heart health. Reduced fibrinogen synthesis. Folks, fibrinogen levels are the top predictor of cardiovascular disease. What else? Increased myostatin gene expression 9.4-fold. Myostatin inhibits heart failure. So now you know the bigger, deeper story on the GHK. Here's a couple of things. If you go to PubMed, you're going to see right here, it says 124 results. There are 13 pages of studies about the GHK. Here's one of them. This has to do with the rapid infiltration and the chemoattraction of the GHK, promoting cell proliferation and angiogenesis. That's creation of new blood vessels. Regenerative and protective actions of the GHK peptide in light of the new gene dating. Go in and read this stuff. So here's the final result. Significant increase of the GHK copper concentration in the blood of the active group was seen in the p-value. You're going to find that out from Dr. Stacy coming up. What is the p-value for comparing changes from day 2 to day 7? So the biggest result was between day 2 and day 7, not between day 1 and day 2. This p-value is lower than the standard to indicate significant improvement or significant value, significant results.
Now, if you could share the X39 results with your heart, how you mentioned how it is very important. Would you mind to share that with everyone very quickly?
Yeah, without the slides, I'll just tell what happened.
Yes.
Okay, I noticed something weird in my heart when I was young, when I was a teenager. I didn't know what it was. I never told anybody. The doctor, when we would go for our little physicals, they would say, "Yeah, you got a heart murmur." And that's pretty much it, because I was a kid and they just told my parents. They didn't know either. They weren't involved in healthcare. Your son has a heart murmur. So when I started using the X39 in April of 2020, I just had this idea. I thought, well, not just because I'm a man where we think more is better. You know, if one's good, 13 would be better. No, I thought if I put one in the normal spot and then right over my heart, maybe I could create change. And I had a knee replacement surgery coming up because I was a crazy skier and broke my leg and tore my cartilage in 1973. And I, all of a sudden, about 4 months after I started using the X39, those PVCs got really regular. It's like every fifth beat, day and night. I'm getting worried because I think, man, what's going on with my heart? This weird stuff. It sounds bad, but it was really actually a good thing. So I made an appointment with a cardiologist, and I'm so glad that I did, because I was able to document something that I had that I did not know I had. I had a stress echocardiogram done a couple of weeks before my knee replacement, and he said, after the test, he said, your heart responded well to exercise. I already knew that. I used to run 40 to 50 miles a week. It didn't bug me when I was running. It was PVCs at rest. And he came in and says, you just have these PVCs in this right bundle branch block. And I said, well, what causes that? And he said, well, you know, we get apoptotic cells everywhere. Those are cells that are killed because they're old, sick, and dying. And we're supposed to replace them, but it's difficult to do in the heart. And the light went on for me, and this is what I thought. I have no proof of anything of it. I have ample stem cells now. The cells are being replaced. They don't know how to beat yet. They're getting the wrong message or something. They're not connected. I don't know. They're all beating at the same time. They're all causing this to beat. So 2 weeks later, my PVCs went away. They're not mine. The PVCs went away. But you know what my first thought was when he came in and said, you have this right bundle branch block? My first thought was, well, I've been using these stem cell activation patches for 4.5 months. Why didn't they go away? Why didn't it heal them? But I was persistent and kept using them. A year and a half later, I had another stress echo done, and the results came back, and it says, right bundle branch block no longer present. 2 different cardiologists have said they've never seen one of those go away. And forever grateful I am, my family, because if you have a right bundle branch block, all-cause mortality goes up because anything you have gets worse. So that's what happened. If somebody, first of all, if somebody judges the patches on 6 months of use or a year, I just tell them my story. Don't give up before the miracle happens, because you're increasing your stem cells. This brain mapping study, it's actually where my board certification is in brain mapping and neurofeedback. And so I hope to give you insight into this. It was done by SciTech Labs. They did a non-invasive scan. It is a non-invasive scanning and subtle energy testing lab. So the effect of the Lifewave X39 patches on the brain, as seen with P3 brain mapping. And we won't get really technical on that, but I'm going to give some basics about it. Principal investigators, you can go back and read all of those. So I want to explain and define for you a few terms before we go and show their actual brain maps. EEG coherence, electroencephalogram coherence. It's a measure of the synchronization of signals between two sites and may be interpreted as an expression of their functional interaction. How are they communicating and sending these electrical messages back and forth? Coherence quantifies the degree of the interaction or communication and shared information between brain sites. Here's coherence. And I put different colors on this side than this side because truly we have two separate brains, kind of, in that there are a lot different things that happen on the left side of the brain compared to the right. And that's what the colors represent. But we have this communication back and forth, coordinating with both sides of the brain. Normal coherence is when there's a normal amount of traffic going back and forth. And then we look at, is there's just the right amount? And then we look at this, more of an explanation, when brain sites are not functioning in an efficient, interdependent fashion, they have too much crosstalk, they are overly connected and locked together. That's hypercoherence. So here's, I show, got the same thing going on the left as the right. It's not supposed to happen that way. Hypercoherence is too much. There's too much crosstalk. It's like a jammed up freeway during rush hour. Nobody goes anywhere. So everything gets slowed in the brain when there's too much. Hypocoherence is when it's called poor intersite interaction, and it's an indication of diminished cognitive efficiency. So here would be hypo. It's like nothing, there's not, I don't have much here and much here, and I don't have enough crosstalk to coordinate all parts of the brain. Because truly, I'm sure you've heard this before, but 80% of some things are processed on the left side, some are processed on the right. But the actual facts are things are processed 80% on one side, but 20% on the other. And the same is true for everything. So, but truly, everything is processed partly by the entire brain. It just needs to be lopsided one side to the other and then cross-coordinated. A little bit different look at it. Hypercoherence. Both sides of the brain are struggling to process information, and they basically try to recruit each other and therefore have more brain cells to do the job in processing the same information. Things are jammed up. Hypocoherence, that's where there's not enough communication between sites in the brain, and it implies cognitive processing. So the study looked at, and move that out. Oops, sorry, gotta go back. The study looked at the degree of hypercoherence and hypocoherence. Where is there too much traffic, and where is there not enough in each individual, and how they improved as a result of wearing the X39 patch. So what were they measuring? And this gets a little technical. I'm just going to make it understandable. So I have to get some things out of the way. There we go. So physical reaction time, we looked at that. How long does it take after hearing something to do something? Trail making. A standard measure of brain function includes measures of psychomotor and visual scanning. Auditory P300, what they do is they send in a sound, an audible sound in headphones, and watch how fast those messages go to the places that perceive that sound. And if it goes too fast or too slow, that's abnormal. This CZ theta beta, just know that if their beta waves are involved in concentration and fast processing, theta waves are more like halfway between sleep and awake, meditation, and memory processing. If your theta waves are too overpowering for your concentration beta waves, it's ADHD, it's ADD, it's aspects of autism and Asperger's. These are the things we treat in my office, and the X39 actually facilitates that. F3, F4, this has to do with there are two of our brain waves that are supposed to be asymmetrically more powerful on one side or the other. They are beta and alpha. When they switch, if alpha switches to the left, more powerful, it's depression. If beta shifts to the right, it's anxiety. Now let's get this thing around. Okay, so that's good enough for that. Hopefully that was understandable. Let's look at some results. So here's what the brain map looks like. And all right, so the red is areas of hyper coherence, overactive brain. I can't focus, I can't process stuff, everything's all jammed up. Blue is hypocoherence. Green is good, good, green, yellow, good. You see from session 1 to session 2, look what happened. It wasn't a straight line to get to session, the third EEG. It actually kind of overcompensated and then had some hypocoherence. So this is why it's important to set someone's expectations correctly. As your brain goes through shifts, and we see this in my office every day, where on your way to something good, something kind of goes a little under functioning, but then it goes back. And that's what the researchers stated at the end of the study. Here's another one. This brain is way overactive. Almost the entire brain was in hypercoherence. But the halfway mark, he just had it in the frontal lobes. And now you start to see some green and some a little underfunctioning. And then the whole brain is working better than this on the left. Here's another one. Way overactive brain starts immediately to start getting better function and communication side to side in this one. And then has, for the most part, green, yellow, blue. But it was only 6 weeks. But this guy had better brain function, better cognitive ability. This one was really interesting because started out in hypocoherence. The entire brain went into hypocoherence before it started shifting. This is a long-term project right here. To get this brain, you see a lot, much better functioning here. But the interesting thing that she said that she was feeling a lot better. And we see that when we used to do these brain maps. We don't need to do them anymore with the new system, but we would see small changes in somebody's brain maps, but huge changes in symptoms. So the conclusion of this study, all participants showed dramatic changes in their scalp topographic maps, reporting the amplitude of the P300 recording. So without getting all that technical stuff, the bottom line is everybody had better functioning brain, less hyperactive brain, overactive brain. And some of you, if you have an overactive brain, I can't shut my brain off at night and I'm having a hard time focusing. I can't process things. I don't remember things. I might have anxiety. I might have depression, ADD, ADHD. The more hypercoherent someone has, the more autistic he or she is. Dr. Stacy and I had a discussion about this, and we truly believe that a rising tide raises all ships, and a better educated group of branding partners, we are going to present ourselves better to the public and stay away from, diagnosing or treating, pretending to treat things. We will keep that in perspective and keep out of the FDA's, list. I almost said the S word list. But you don't want to do that. And so we try to help people understand how they work and what we can say, what we shouldn't say.